Bioequivalence studies by adaptive designs is the topic for present video, and adaptive designs are the designs which allow or which are flexible for performing the bioequivalence studies. Welcome dear viewers to this informative topic, and let's start with the introduction. Adaptive word or adaptive meaning we should understand first before going to the next slides. Adaptive means having a ability or tendency to adapt different situations. That means the adaptive word gives you the idea about to adapt or be flexible to accept some changes. Adaptation is the process of changing to better. Adaptive designs in the bioequivalence is a clinical trial design. This may be used in the adaptive design may be used in the bioequivalence or clinical trial designs that allow some modifications or some changes to the trial after it has started, while still maintaining the trial's validity and integrity. The goal of the adaptive design is to make clinical trials more efficient, flexible, and faster. By adaptive designs, the cost can also be saved. So these are the cost-effective designs. One thing I can tell you that the bioequivalence studies or clinical trial studies are performed with the protocol, and if the protocol is changed, that time it comes into the category of deviation of the protocol. But the adaptive designs are such designs in which the would-be changes are reported in the protocol. That means the protocol is written in such a way that the adaptive design is formed, or the would-be changes or planned changes are given in the protocol, and that's why these designs are more adaptive, flexible, faster, and cost effective. So the meaning of this exact design you will understand in the upcoming slides. So be with me till the end of this video. First of all, we will see what is bioequivalence. Bioequivalence refers to the comparison between two pharmaceutical products, and generally these are generic formulation or generic product and the reference product, to determine whether they release the same amount of active ingredient into the bloodstream. Also, you can say that bioequivalence is the comparison of bioavailability, including the rate and extent of absorption or pharmacokinetics between the reference product and the test product. Now coming to the importance of adaptive designs. Why adaptive design came into picture for bioequivalence studies and clinical studies. Adaptive designs offer flexibility during the clinical trial process, allowing modifications based on the interim results, optimizing resources use, and reducing time to decision making. The example I will give you so that you will understand the terminology from this slide onward. For example, for bioequivalence, you have taken 100 subjects and you have made two groups of 50 subjects in each group. Now, if you are not following the adaptive design, you have to perform bioequivalence with 100 subjects. And if you reduce number of subjects, then it will be a protocol deviation. But in adaptive design, you can write that I am using 100 subjects, but in the group of 50-50 each by making the two groups. And if bioequivalence data gives the idea about bioequivalence proof or demonstration of bioequivalence in the 50 subjects only, then I will evaluate the data and I will not go for the testing in remaining 50 subjects. That means you have chance or you are flexible to change some reported or flexible changes during the trials. That means you are allowing the modifications in the design based on the interim results. And that's why you can optimize the resource use, you can reduce the number of patients or subjects, and that's why you can reduce the time for making the decision. So this is the importance of adaptive designs. You can also make adaptive design saying, if bioequivalence is failing in 50 subjects, then I will go for another 50 subject testing. Now I hope you might be clear on this, and now we will go by other slides in detail. What is adaptive design? Trial design that allows adjustment based on the interim analysis. Modifications might include sample size changes, stopping early success or failure, and other adaptations. So based on the output of the design or study, you can modify. Types of adaptive designs. So sample size re-estimation is the one of the design. Then group sequential design is another type of design. Bayesian adaptive design is the another design, and early stopping or success utility design is the type of adaptive design. So mainly here I have enlisted around 4 types. There may be some other types as well. Coming to the US FDA guidelines for for adaptive designs, FDA bioequivalence guidance provides you the information regarding adaptive designs must be pre-specified in the study protocol. So it is very important to pre-specify your would-be changes. Modifications must not compromise the study's integrity. This is very important. Statistical considerations, importance of controlling type 1 error, use of statistical methods, gatekeeping procedures, or Bayesian methods to adjust the designs. Interim analysis or interim analysis, real-time analysis to inform changes in sample size, dosing schedule, and other trial parameters. So this is the overview of the adaptive design. You can do some changes in sample size, in group sequence, and the other designs you can change. And based on your interim results, these pre-specified changes can be done without compromising the integrity of the study. FDA guidelines are there for sample size re-estimation, sequential design, ensuring study integrity. Sample size re-estimation involves adjust sample size during the trial if variability is higher or lower than anticipated. So you might be knowing about this. If variability is more, you can increase the subjects. Or if it is lower, then you can stop the study at that point. Sample size re-estimation is aimed at ensuring the adequate power for detecting bioequivalence. Sequential design, staged testing where B can be assessed progressively. Early stopping can be implemented for futility or success. Ensuring study integrity, use appropriate statistical techniques to maintain the validity of the study even with adoptions. So study integrity is must. Now see the ICH guidelines on adaptive designs. ICH E9 guideline provides the statistical principles. It is a broad guidance on clinical trial designs including adaptive designs. Ensure statistical integrity and robustness of the trials. Pre-specification of design elements. All adoptions, sample size changes, stopping rules, etc. must be pre-specified in the study protocol. Transparency in the planning phase is a key. So as I have mentioned earlier, the changes should be pre-specified. It is not like that you started the study and then you got the positive results or negative results and then you are going to change the protocols. It is not like that. The changes or would be changes, adaptive changes, adaptive design should be pre-specified and it is properly planned. ICH guidelines give you information for maintaining statistical integrity, ensuring that the adapters do not inflate the type 1 error. Error of type 1 should not be there whenever you are performing or planning for the adaptive designs. Use rigorous statistical methodologies such as Bayesian or classical methods to control error rates. The type 1 error rate should not get compromised because of the adaptive design. Regulatory compliance adaptive trial design must be aligned with regulatory standards. Adoptions need to be justified, documented, and approved by the regulatory bodies like FDA, EMA, and other country-specific regulatory authorities. See, now coming to the key types of adaptive designs for B studies. What are the types? What changes you can do? These are the key types of adaptive designs. First is sample size re-estimation. That means you can adjust the sample size. Adjusting sample size based on early data to ensure robust results helps to avoid underpowered studies or unnecessary resource use. Group sequential designs, multiple analyses at predefined points to assess bioequivalence early, allows for stopping the trial early if results are clear. Whenever it is failure or success, that time this group sequential design can allow for stopping the trial early if the results are clear and robust. Then Bayesian adaptive designs, use of proper knowledge and existing data to inform decisions during the trial. Bayesian models update the probability of success as new data comes in. So adaptive design with Bayesian provides you the information with prior knowledge and existing data for taking the decision during the study. Early stopping for success or failure or futility. Stopping the trial early if bioequivalence is clearly achieved or if it is deemed unlikely to be achieved. It saves the time and resources. If you come to the point that you know the bioequivalence is passed and there is no need to go ahead, or if you come to know that this product is not going to pass the bioequivalence test or study, then you can stop there itself. Now see the statistical considerations in adaptive designs. Controlling type 1 error. It is necessary to prevent inflating the probability of incorrectly rejecting the null hypothesis. That means false positives. Adaptive designs require special methods like gatekeeping procedures, Bayesian adjustment to control this risk. And risk is what? You are rejecting the null hypothesis. You need to control type 1 error. So these statistical considerations should be there in the adaptive design. Interim analysis. Data from the trial is analyzed periodically and adjustments are made accordingly. The design should be robust enough to account for these changes while maintaining the trial integrity. The adaptive design and the results should not lead to failure to maintain the trial integrity. And the design should be done in that way. There is no failure in the trial integrity, or you are not going to harm the trial integrity. Advantages of adaptive designs in the B studies. So certainly there are many advantages, like it allows for faster decision making in the real-time data with using the real-time data. So it improves the efficiency. It improves the utilization of resources. Resources are used more efficiently in the study designs. Example, adjusting sample size based on the variability. Cost savings. Fewer patients may be needed, reducing cost, reduced trial duration. So cost and time can be saved by adaptive designs. Ethical considerations. Reduces unnecessary exposure to treatments or placebo when it is clear that bioequivalence has been achieved or it is unattainable. It is very important to understand that the adaptive designs improves the efficiency, reduce the cost, and also these are the designs which reduce unnecessary exposure of the medicines to the patients or the healthy volunteers. Now we'll see what are the challenges and considerations. As in adaptive designs, the decisions are made to reduce the time, cost, and also to get the data in a short duration or in sufficient time. So there are many challenges. First challenge is complexity in implementation. Second challenge may be regulatory approval challenge, maintaining the validity of the trial and data. These are the challenges. Other challenges are also there. So first is complexity in implementation. It requires careful planning and execution, especially in the statistical methodologies. It can be resource intensive and may require specialized expertise. So expertise is required. It requires proper planning and execution. That's why it becomes little bit complex. Regulatory approval. Regulatory approval should be there for adaptive designs, and adaptive design must be fully justified and approved by the regulatory bodies like FDA, EMA, and other country-specific regulatory bodies. Clear documentation and communication with regulators are crucial. Maintaining validity, ensuring that the integrity of the study is maintained even with modifications. Discuss how the study meet its bioequivalence endpoints with fewer resources. Sometimes, because of the reduction in the sample size, there may be uh, variation in the data. There may be statistical errors. There may be problem of sufficient power of the study. So these are some of the challenges which are required to be evaluated critically. And in summary, we can say that adaptive designs offer flexibility and efficiency in bioequivalence studies, in clinical studies, but they require careful planning and rigorous statistical methods. FDA and ICH guidelines provide frameworks to ensure that adaptive designs are used appropriately without compromising the integrity of the trial. Benefits of the adaptive designs include enhanced efficiency, reduced costs, faster timelines, and ethical advantages. Challenges of the adaptive designs are complexity in the design, expertise is required, regulatory approval should be there, and maintaining the statistical validity, reducing the errors. So these are the important points for the adaptive designs for the bioequivalence studies. I hope this video will help you to understand these bioequivalence designs. Thank you for watching the video, and let's meet you soon with such kind of informative videos.